AS Level Biology 9700, Paper 1, Variant 2, May June 2025. Students, today we are going to discuss from question number 1 to question number 10. And so I'm going to start with question number 1, which shows possible uses of an eyepiece reticule. An eyepiece reticule is a small glass disc with an edged scale that is placed inside the microscope's eye. So it is used as a measuring tool. So they have given two statements regarding the uses of an eyepiece reticule, and we have to find which statement is correct and which option is correct. Comparing the diameter of two cells viewed with a microscope, magnified objective lens. So, on the other, calibrating a stage micrometer viewed with a 10 objective lens. So here power 10 objective lens. So here you can see comparing the diameter of two. Actually, this eyepiece reticule is used to compare the cell diameters, because with the help of this, we can find out the diameter of the object. So it is possible to use an eyepiece reticule to compare the diameter of two cells. So reticule scale is fixed. So you can measure each cells in reticule scales and then compare those measurements directly. So it means this is the possible use. So we are going to take with it. Second, calibrating a stage micrometer viewed with the power 10 objective lens. So this statement or this possibility is that we calibrate the eyepiece reticule with the help of stage micrometer. So what is stage micrometer? Stage micrometer is also a scale which is used to calibrate the eyepiece reticule. So this statement is not correct. So this is not the possible use of an eyepiece reticule in simple words. So according to this, A statement, no. Again, B can be the correct option according to our findings. C again, no. D, no. In which, so these are not the correct options. So only B is the correct option, because the eyepiece reticule is used to compare the diameter of two cells when we view with the power of 40 objective lens. So on the other hand, we calibrate the eyepiece reticule with the help of stage micrometer. The eyepiece reticule is used to calibrate a stage micrometer. So we are going towards the question number 2, and question number 2 is, which statement about a light microscope is correct? So as the distance to see two points as separate points decreases, the resolution also decreases. First of all, you must know what is the resolution. Resolution is the ability of a microscope to distinguish two separate points. That is, the separate points which are very close to each other. So very close to each other. If the resolution is high, we can distinguish between these 2 points. So as the distance to see 2 points as separate decreases, the resolution also decreases. This is the, you can say, incorrect statement, because if the distance is decreases, it means the resolution is increased. So bacterium 0.2 micrometer in a diameter will not be visible if the resolution is 220 nanometer. 220 nanometer is equal to 0.2 micrometer. Because if we divide 220 nanometer with 1000 to convert it nanometer into micrometer, so it will be equal to 0.2. So a bacterium in 0.2 micrometer will not be visible if the resolution is 0.2 micrometer. So this is the correct statement. because the resolution limit of a standard light microscope is generally accepted to be around 200 nanometer, because the shortest wavelength, and according to the formula, the theoretical maximum resolution should be the half of the wavelength. So according to this, I would like to say uh, that statement is correct, because the resolution of the microscope is 220 nanometer. It can only distinguish objects that are 220 nanometer or further apart. So according to this statement is correct. The third statement, two membranes that are less than 300 nanometer apart will be visible as two separate membranes if the wavelength of light is 600 nanometer. So the two membranes that are less than, so they will be considered as, so this is not correct, because a wavelength of 600 nanometer would have a theoretical resolution limit of approximately 300 nanometer. So wavelength 600. If the wavelength is 600 nanometer, so resolution limit, it will be 300 nanometer. So if there is less than 300 nanometer, so there will be this membrane will appear as a single blur line. So they will not be visible as two separate membranes. They are saying that will be visible. No, it will not be visible. So if it is less than 300, so it should be 300 nanometer, then it will be visible. So according to this statement, it is incorrect statement. Now the next statement is the resolution will improve with visible light of a longer wavelength, such as red light. Again, this is incorrect statement, because the resolution improves as the wavelength decreases. The resolution will improve as the wavelength decreases, but here longer wavelength. So if wavelength is increasing, the resolution will decrease. So again, this is not correct statement. Only B is the correct option. Question number 3. And question number 3 is the electron micrograph shows a cell. What is the actual maximum length of the cell? First of all, you must know what is the formula for the magnification. I would magnification is equal to image length over actual length. So we have to find out the actual length. So actual length is equal to image length over magnification. Magnification has been given. We have to find out the actual length. So actual length will be, so we have to measure the distance, largest distance on this image, the longest distance. Let me draw it again. Actually, with very difficult, because this is a touch screen, very difficult to draw with the help of a scale. So I'm trying my best, just understand. So this distance, we will measure the distance between these 2 points. So it is equal to 7.3 centimeter. So we have to convert 7.3 centimeter. First of all, 7.3 centimeter is equal to, you have to use the calculator over here. So 7.3 is equal to 73 millimeter. Convert 73 millimeter into, by multiplying it with 1000, it will be equal to 73,000 micrometer. So actual length is equal to 73,000 over this magnification 108,123. So I'm going to divide. So it is equal to 0.675, which is nearly equal to 0.676. So according to this, the B is the correct option. Now we will move towards the next question. Question number 4. So question number 4 is, what is the actual, question, the table compares some biochemical molecules of P, Q, R, and S. So they have given us 4 molecules. And these 4 molecules can be the chlorophyll, DNA, RNA, and peptidoglycan. So we have to go with them. So first of all, chlorophyll is present, and DNA, RNA, peptidoglycan. So first, chlorophyll is present. Chlorophyll is present in chloroplast. Okay, so chloroplast, this. So chlorophyll is present in chloroplast. Secondly, DNA, RNA, they are also present in chloroplast. So with B, we will go chloroplast. Second is Q, which is no chloroplast, DNA, RNA. Yes, DNA, RNA, they are present in mitochondria. So Q is mitochondria. And then R, DNA, RNA, and peptidoglycan. You know, the cell wall of the bacterium is made up of peptidoglycan. So it can be a bacterium. And then RNA. So it is a virus. That is, RNA is present. Chlorophyll, DNA, and peptidoglycan all are absent. So it can be a virus. So according to these correct, we can find that B is the correct option, in which P is the chloroplast, which contains chlorophyll, DNA, RNA, and the Q is the mitochondrion, which contain DNA and RNA, and R is the bacterium, because it contains peptidoglycan beside DNA and RNA. And then virus contains RNA, no chlorophyll, no DNA, and no peptidoglycan. So B is the correct option. Now we will go with the next question. Question number 5. And question number 5 is, which cell structures contain enzymes and are enclosed by a double membrane? So first cell structure which they have given is nucleus. You know, the nucleus is the control center of the cell. So it has double membrane. It has double membrane. This is confirmed. It is enclosed by, the double membrane, which is called as nuclear membrane. So it contains of, you can say, enzymes, because uh, the enzymes, it has to perform the functions, as for example the DNA replication. So enzymes are present, and it also has the double membrane. So nucleus, the uh, next is mitochondria. And mitochondria, the respiratory center. So powerhouse of the cell, sometimes we say that it is power, and it is the respiratory center, and it has double membrane, inner membrane and outer membrane. Inner membrane, has many folds into it, which are called as cristae, so which increase its surface area. And the mitochondrial matrix is rich in enzymes, as for example Krebs cycle, oxidative phosphorylation, glycolysis. All these different functions takes place in the mitochondria. So the mitochondria both have the double membranes and enzymes. Then the next one is organelle, is the chloroplast, which is only present in the plants, and photosynthesis takes place over here. So chloroplast also has two membranes. So it is membranous structure, and then it has, because the formation of carbohydrates take place over here, so it also contains the enzymes for the process of photosynthesis. So all of these organelles contains enzyme, and they are enclosed by double membrane. According to this, A is the correct option. B, C, and D are not the correct options. Now we will move towards the question number 6, and question number 6 is, what is the order of size of cell structures? So they have given us 4 structures, and we have to arrange these cellular structures. So I will, first of all, I'm going to write that the centrioles, centrioles are 0.5 to 5 micrometer size. Lysosomes, wait a minute. I have, these are nucleolus. Has 0.5 to 5 micrometer. Lysosomes have 0.1 to 1.2 micrometer. And then centrioles, they are 0.4 micrometer. And then ribosomes, 25 nanometer. So according to this arrangement, as you know, but their size vary. Their size can be varied, so they can be between these. So according to, if we go with them, the nuclei is the, if we compare all of these, nuclei is the largest, and from nuclei, the lysosomes are at second place. Nucleoli or nucleolus, the nuclei is the plural of nucleolus, and nucleolus is used for the to synthesize the ribosomes. So it means nucleoli is the largest and it is present in the nucleus. Lysosomes are the second organelles which are largest in size as they have to um, contains many enzymes. So they are on the second number. Then centrioles. You know the centrioles are rod-like structure which are used for the cell division. So they are at the ribosomes are smallest in size. They are smallest in size. They can pass through the nuclear envelope. They are attached on the endoplasmic reticulum. So they are as at fourth place. It means according to this arrangement, C is the correct option. A, B, and D are not the correct options. Now we will move towards the next question, and next question is a sample of a solution of sucrose tested for reducing sugars remains blue, but when another sample of the same solution is tested for non-reducing sugars, the solution turns red. Reducing sugar, which is used to test the monosaccharides, glucose and fructose. So, reducing sugar, Benedict is also called as Benedict test, which is used to find out the presence of monosaccharides or glucose or fructose into a given sample. Whereas the non-reducing sugar is for the polysaccharide, as for example, it can be for sucrose and other polysaccharides. So we have to find out the correct option according to the given statement, which explains these results. During the non-reducing sugar test, an acid hydrolyzes sugar to glucose and fructose. Yes, when we perform the process of non-reducing sugar test, first of all, before non-reducing sugar, we use the acid, we heat it up, and it will break down the polysaccharide or disaccharide into monosaccharide. That is, the sucrose will be broken down. Here they have given us sucrose. So sucrose, when we use acid, it will hydrolyze the sucrose into glucose and fructose. So this is the correct option. During the non-reducing sugar test, sucrose molecules condense into polysaccharides. There is no formation. There is the breakdown because we have used the acid which will hydrolyze. So first statement is correct, second is not correct. The reducing sugar test converts sucrose into glucose and reducing sugar is used to test the monosaccharides, not to dis or polysaccharides. So this is not correct. Again, hydrolyzes, so it will not hydrolyzes. They are only used for monosaccharides. So only A is the correct option. Now we are going to move towards the next question number 8. And question number 8 is which molecule contain at least two double bonds? They have given us three. Saturated triglyceride. Saturated triglyceride means there is no double bond. There is no double bond in the saturated triglycerides. There will be no double bond. Hemoglobin, again, it don't have any double bond. Collagen, they don't have the double bonds inside. So according to this, so saturated fatty acids, no double bond. There is no double bond in hemoglobin as well. No double bond will be there. Collagen is fibrous protein. Again, it is a primary structure, it has a long chain, so there will be no double bond inside. And then I come to the hemoglobin, and hemoglobin is a globular. This is fibrous, and this is globular. So all of them will not contain any double bond. So according to this, D is the correct option. No one contain any double bond. So D is the correct option. So I'm going towards the question number 9. Question number 9. So question number 9, amylose, amylopectin, and glycogen are all polysaccharides. So enzyme X removes one one maltose, which is at a time from the ends of a polysaccharide molecule by hydrolysis of alpha 1 4 glycosidic bonds. So which row shows how completely or each of these each of these uh, polysaccharides is composed of the same number of monomers before hydrolysis begins. So largely complete. So first of all, I'm going with amylose. Amylose is unbranched. It is a straight chain. Amylose is a straight chain, unbranched, in which 1,4 glycosidic alpha 1,4 glycosidic bonds are formed. So amylopectin is branched, in which 1,4 with 1,6 both type of glycosidic bonds are present. And whereas the glycogen is highly branched, it is highly branched. So it means which is highly branched, it will least completely hydrolyzed, and then which has branch structure, and then which has no branch structure. So according to this, amylose is no branch structure. So A and B are not the correct option. In C and D can be the correct option, but the on second number there should be the amylopectin, because amylopectin is more branched structure, but amylose is not a branch structure. So according to this, glycogen and then amylopectin and then amylose, they will be most completely hydrolyzed, and least will be the glycogen. So D is the correct option. Question number 10. Which property of water enables sweating to be an efficient means of losing heat from the body? So latent heat of, first of all, you must know what is latent heat of vaporization. So the latent heat of vaporization is the amount of energy which is used uh, to convert the liquid into a gas, or water, it is to convert the water into vapors. So water enables sweating to be an efficient means of losing heat from the body. So it can be the correct option. The specific heat capacity. Second one is the specific heat capacity. So the specific heat capacity is allow water to absorb a lot of heat without a significant change in its temperature. So heat capacity of water is high. So this is not the correct option. Then we go with the third. Each water molecule can form a hydrogen bond with 4 water molecules. This property does not matter the sweating. So this is not a correct option. Next one is the forces of cohesion and adhesion on water molecules are very high. Molecules move with the help of this cohesion and adhesion movements when the water has to move during the transpiration pull. So again, this is not related with the sweating. So I would like to explain this for you. That is the as the sweating, I will go with first of all how sweating cools the body. So when we get hot, our bodies produce sweat, which is mostly water and release it into the skin. So the sweat absorbs heat from the body's surface. So the absorbed heat provides the energy needed for the water molecules to overcome their hydrogen bonds and change from a liquid to a gas. So according to this, A is the correct option. Whereas the we also say can say that this process is also called as the evaporative cooling. So our body, so A is the correct option. So according to the specific heat, it capacity of water, if it is high, so but this property of water will not allow water to absorb a lot of heat without a significant change in its own temperature. So which is important for moderating body temperature, but it is not the direct mechanism for cooling through sweating. So this is not the method which can help the you can say to lose the heat from the body. So B, C, and D are not the correct options. So it was students, it was all about from question number 1 and 10 to 1 to 10. Please like, share, and subscribe my channel. So in this way, you will get notification when I will upload the other videos of paper 1, paper 2, paper 3, 4, and 5. Thanks for watching.